Okay, I think we'll get started then. Good morning, everybody, and welcome to the first of a double header that we have in the Biophysical Techniques lecture series, in which we introduce the topic of NMR spectroscopy in the context of the kind of work that we do here at the LMB. LMB has an exceedingly well-equipped NMR facility. It's one of the largest in the UK, and certainly the quality of the equipment that we have access to is second to none. What I want to do today is go through a brief introduction to the scope of the kind of work that we do. I want to look at what is the basis of the technique, what is it that, what properties of a molecule affect an NMR spectrum, and therefore we can work backwards, and by measuring NMR spectra, we can infer behavioral properties of the molecules in solution. And then on Thursday this week, Jane Wagstaff is going to give an overview of some non-routine advanced applications that have been done in the NMR facility here in the last couple of years. So let's first begin. Oh, it's not going to start. My slide advancing for some reason. There we go. There we go. Slick as a start. Right, the first place of contact then, if you want to know anything about the NMR facility here and getting in contact with anybody here, is go to the internal web page, and you can find under the Scientific Facilities tab, we can see who it is that you are, it's your points of contact within the LMB. So that's your first point of call if you want further information. And then there are various people around for you to collaborate with and get advice, detailed advice about the physics involved in the NMR experimentation. So where are we physically? Well, we invented social distancing. So from the year 2000, we erected a new building that is specifically designed to house NMR equipment. NMR magnets aren't very friendly to some of the scientific equipment in neighboring rooms. And similarly, we have interference from other equipment on ours. So we put in a purpose-built NMR facility that is something like 5 minutes walk from the main LMB building. I should note at the top here that the word nuclear magnetic resonance spectroscopy is changed to magnetic resonance spectroscopy if you're on a clinical site where you have patients around. The nuclear word, the dreaded N word, is disfavored. So instead of calling it NMR in a clinical setting, we refer to it as MRS for magnetic resonance spectroscopy, as opposed to MRI, which is the nuclear magnetic resonance imaging facilities that you have in hospital sites. So we are in a separate building to keep us nice and free from interfering with other techniques. We have 7 NMR spectrometers available to us on the site, and we also have access to higher field facilities elsewhere in the UK, particularly at the Crick Institute in London. We have an 800, a 700, 2 600 megahertz spectrometers, and a 500, all housed in the MRS building. You are not expected to be able to run this stuff yourself. NMR has an enormous amount of technical detail involved, and for that reason, it tends to be that you collaborate with staff in the NMR facility who will see you around any problems that you have with getting information off the equipment. We also have 2 unattended open access NMR spectrometers in the LMB main building, 2 400 megahertz machines that are used to support the organic chemistry efforts. So what is it that we've invested so heavily in NMR for? What is NMR going to give us? Well, the primary application of NMR spectroscopy is looking at the composition of molecules. So every chemistry department certainly in this country, and pretty much most of them in the world, in universities, will have an NMR facility that is used to determine the composition of small molecules after synthesis. But we can also use it in a wider context to look at the shape, the 3-dimensional shape, and the formation of complexes, intermolecular associations. So in a biomolecular context, the main applications that we have is, first of all, to look at solution structure and internal dynamics. Note that we're looking at the native structure of the protein in solution, which is one of the big advantages. So it should be free from crystal packing artifacts. We can verify that we're not getting perturbation of the structure due to crystallization conditions. But we also have a look into the internal dynamics of the molecule, which NMR provides a handle on, and we'll say a little bit about that today. I should say that we can also see structure in proteins that aren't folded. So we can look for the presence of residual structure in intrinsically disordered proteins. And we can look at molecular interactions. NMR is one of the gold standard techniques for characterizing the binding interfaces and the presence and the affinities that you get between different receptors and their binding partners. We can produce high resolution models of associated complexes, usually experimental data-driven approaches. We have the principal advantage that NMR gives over other forms of spectroscopy is that it provides a separate signal for every nucleus, every atomic nucleus in the molecule. And therefore you have site-specific resolution as to what is going on inside the molecule. It's a crucial advantage of NMR spectroscopy that we'll come back to again and again. So we are able to identify the locus of any changes and perturbations in structure and post-translational modifications, for example. We can look at quantifying various biophysical constants, such as diffusion rates, individual hydrogen exchange rates, looking at stability of folded proteins and the radius of hydration. NMR is quantitative, so we can get equilibrium constants, dissociation constants of binding affinities, pKa's, and kinetic rate constants. So we can produce all sorts of biophysical data from that. NMR is one of the principal tools for lead generation in the pharmaceutical industry. So every major pharmaceutical company has had some interest in fragment screening libraries and fragment screening programs. And NMR forms a central part of the small molecule screening for drug discovery. And we shouldn't forget that we're not just able to look at the macromolecules themselves. We can do the direct observation of substrates and small molecules to look at substrate turnover and metabolic pathways in situ in intact cells. So that's the basic scope. The sort of things that we're doing sort of day to day is we are contributing to the picture and providing a mechanistic view of how large complexes assemble. Something like this from Marianne Bientz's work is a multi-protein assembly that is way beyond the size of direct observation by NMR spectroscopy. It's a transcriptional activating complex, signaling complex involved in cell replication, but where NMR can contribute very valuably is the direct interaction between individual domains. So we can characterize which are the domains that are interacting with each other, at what stage, and we can look at the structural basis of the interaction between these. So we get the contact surfaces. We can look at both stable and transiently associating complexes. And crucially, when we're looking at these, they're free from any crystallization packing artifacts. So NMR isn't going to be a panacea for everything. Okay, it's an extremely powerful technique, but there are some conditions where it starts to face challenges. First of those is the ability to resolve planes of symmetry. Now, that's not always a disadvantage. If we look at, for example, fullerene molecules, C60, one of the key features of determining the structure of this molecule is that every carbon in a C60 molecule has the same environment, magnetic environment. So you get a single NMR signal for 60 carbons, which is telling you an awful lot to constrains how you would do model building in order to make up something that has that form of symmetry. In our case, the sort of place where it becomes extremely challenging is when we have domain swapped dimers, for example. This is a homodimer, again from one of Marianne's projects, where we have one element from the monomer is swapped over into an equivalent position in the neighboring subunit, and these occupy a plane of symmetry. They exhibit a plane of symmetry down the middle, and we're very challenged to resolve the two parts of the homodimer in this context. There is a way in which you could possibly consider having a mixed isotopic labeling strategy where you try to label one of these partners where it's not labeled in the other. So it may be possible to break down the symmetry in some way, but it would be very challenging to identify information from both parts of the homodimer in this case. Heterogeneity, NMR is able to look at mixtures of compounds, and you can look at cytoplasmic contents and pick out individual components by NMR. But in terms of the structural context, when it comes to structural analysis, we really require exceedingly pure monodisperse and preferably single conformers, although we can look at conformational exchange processes. But if the sample isn't heterogeneous, then we start to struggle for the resolution in NMR. Sample quantity is always a disadvantage for us. NMR is inherently a very insensitive technique. We'll come on to the reasons why, but it means that we require, as a sort of minimum, as a ballpark figure, we're going to be looking at solutions of 20 micromolar plus. That's really the lower limit for detection in most of our work. And there's a minimum sample volume of 280 microliters. That's quite a lot of sample compared with some biophysical techniques. NMR signal quality degrades as the molecular weight of the complex goes up. Essentially, as the molecule starts to tumble slower, the quality of the signal is degrading. And so NMR is really more suited to analyzing individual isolated domains rather than looking at multi-domain complexes and full-length proteins. Often applied to single domains rather than multi-domain assemblies, it says there. Low spin density. So something like DNA, for example, when you're looking at proteins, you have a huge amount of number of spins to work with to get information from. But in DNA, you have a sparsity of the contacts. You have a very low spin density. And this is making NMR, structural NMR, a lot more challenging a problem. And speed. In the end, NMR is a time-consuming, laborious process. In terms of comparison of the structure determination process between NMR and crystallography, crystallography wins hands down every time. Within sometimes a few hours, if not maybe a few days, very often once you have the data collected, crystallography will have a finalized structure. NMR will take months to do a formal solution structure. If you need the information, you really want the information, it's worth chasing it. But if there's a chance that you can crystallize the molecule instead, you are much better off chasing crystallography to get the structure out. There are automation routines out there yet, but it is our view in here that they're not reliable enough at the moment to produce publication quality structures without having to do a formal solution structure anyway. It's important for us to say that NMR sees everything that's in the solution. So it sees every state, every individual state of the ensemble, but it doesn't necessarily resolve those. So if we look at solid state methods, for example, cryo-EM can very often pick out different conformational states and resolve classification into the different states. Excuse me. Crystallography tends to be a snapshot of single static states, but what NMR sees is an overall moving picture, and it sees all of those. In order to resolve what the individual contributions are to those, you need to use NMR in combination with other methods, with computational methods, and you need to validate the computational methods against the NMR data. You can quantify both the extent and the rates of motion that are in there, but resolving individual conformations can be sometimes exceedingly challenging. If you were to ignore the information completely and try to interpret the NMR data in terms of a single structure, without going to the whole ensemble, you can result in an erroneous, what's called a virtual conformation, one that's not actually populated in physical space. So we have to be sufficiently cautious about the way in which we treat the dynamics and the presence of the ensemble. This is something that's a slide from Jane's talk later in the week. We've decided to split the talk, that she will do more of a focus on the dynamics, but I am just going to introduce the concepts that we have various processes that are occurring inside all biological macromolecules. And they are occurring on a range of different timescales, okay? And suitably, we have a whole plethora of different NMR techniques that we can use to probe the motions on the different timescales. And what Jane will go into next week, later in this week, is some examples of where we can apply some of those. We don't need to go into the details of how all of these work today in the introduction. So what is NMR about? Where does the signal come from? Well, the basic starting point is that a certain selection of nuclei have the property of a magnetic dipole, a nuclear magnetic dipole. And that has a north and a south pole. And if we place this sample inside a strong external magnetic field, then we will generate two different states, okay, of separate energy. The two states are determined by the quantum mechanical selection rules. When my preference for today is, as we're introducing this, we're not going to look into the details. We're not going to come in from a physics point of view, from a physics background, and look at the mathematical derivation of the process. We'll just look at it in terms of very pictorial, cartoon-like representations as a starting point. So what we get is two different orientations, a lower energy state and a higher energy state, according to whether the spin is aligned with or against the magnetic field as an average. Okay. And certain nuclei possess this property, and they include the most abundant nuclei of hydrogen, which is the proton. The single H1H is just a proton on its own. So we use the terms interchangeably. Proton NMR and hydrogen NMR is essentially referring to the same thing. We have also an isotope of phosphorus that we can look at. The 31P is there at more or less 100% natural abundance, as is fluorine. F19 is also there at a high abundance. The NMR active isotopes of carbon and nitrogen could also be brought into play very usefully in NMR studies, but they're there at very low natural abundance. So if we want to introduce carbon and M15, we need to specifically isotopically label the protein by expressing the protein in a medium that contains C13 glucose and N15 ammonium chloride as the sole carbon and nitrogen sources for the bacteria to replicate on. So we need to put those in specifically if we want them there. Okay, and that's the kind of stuff that is routinely done within LMB. So the energy difference between the two spin states is linearly related to the strength of the magnetic field that we put it into. Right? So the stronger the magnetic field, the greater the separation of this. And the signal that we measure at the end is dependent upon the difference in energy level populations between the two. They are almost exactly 50% populated. In fact, there's only about, in a 500 megahertz magnet, there's an excess population of about 40 spins in a million in the upper state compared to the lower, in the lower state compared to the upper state. And it's the difference in population that is responsible for the strength of the signal that we're going to see. So this is why NMR is inherently insensitive, is because we only have a very slight excess of population in the lower state over the upper state. So the difference in energy level there is about 10 to the minus 25 joules. It's an incredibly small gap, and which is why we have almost equal spin states. So different spectrometers, what is worth just saying very briefly, what's the relative merits of going to different field strengths. If we go to increased field strength, we get an increased sensitivity. Basically, you increase the polarization difference between those two energy levels, and you increase the signal dispersion. You can get some favorable relaxation rates in there, which I won't say a lot about at the moment. The main disadvantage is the cost. For every 100 megahertz in frequency that you go up, it doubles the cost of the spectrometer. You also have to find different buildings, separate buildings to house these things in, because the magnetic fields, stray magnetic fields, become too anti-social to house within areas with other scientific equipment. The basic take-home message from this, though, is the stronger the magnet, the better. Okay. The business end of the NMR spectrometer looks like this. This is a part that you would never see inside the spectrometer, but what we have is a sample in solution, right? So we're looking at, for us, it's always aqueous solution. The sample depth is 4 centimeters, and it sits inside the coil of a detector probe, okay? The detector probe is set up so that it will detect any magnetization that is spinning inside this, within the confines of the coil. Okay, so the basic sample conditions that we would have is fixed sample volume of 550 microliters. The concentration range that we would go to is we wouldn't go much below 20 micromolar. We wouldn't normally encourage going above 500 micromolar, because at that point you start to run into high likelihood of protein aggregation. So we would normally recommend people keeping at no more than about 500 micromolar. And we have a temperature range that is moderately usable for biological studies. Okay. You note that the sample does not get in contact with the machine directly at any point. It stays within the sample tube, and so in principle you can recover your sample at the end and take it back to do other things with it. So inside the spectrometer we have the precessional orbit. This spin is the bar magnets are spinning around and producing a net bulk nuclear magnetic moment, and that is strong enough for us to detect the rotation of that in the receiver coils. Okay, as that happens, what it generates is a sinusoidal oscillating electrical signal in the receiver circuit. Right? That is the basic NMR signal that we are going to be working with. So somehow we have to convert that to a useful spectrum. And the idea is that if we take the process of Fourier transformation, which is not a complicated process, we can take the time domain signal that we measure, pass it through the Fourier transform algorithm, and what you get is a delta function. It has a discrete single frequency. It's a single valued function describing what the frequency component of that wave is. Okay. In practice, what we have is a damped cosine wave, sinusoidal wave, which is decaying with an exponential function, and the exponential time constant is this value t2. If we pass that through the Fourier transform, we get the so-called Lorentzian line shape that has critically both an amplitude that is defined and an intrinsic line width, which is depicting the uncertainty of that center frequency. Notice that the center frequency itself is exactly the same, but now we have the associated line width of that. The line width is inversely proportional to the rate of decay of that sinusoidal wave. So if we look at that relationship between line width and the rate of decay, you can see that for a large molecule that tumbles very slowly in solution, for a high molecular weight protein, for example, we might see a short T2 time. So relaxation, the process at which this system returns to equilibrium following an excitation pulse, that relaxation occurs very efficiently for slowly tumbling molecules. And if you do a Fourier transform with that, you'll see you get a broad line width. For a small molecule, where the signal is lasting very much longer because relaxation is so much less efficient, you'll get a very much smaller, narrower line width. This is the reason why NMR starts to struggle as we go up to slowly tumbling molecules. As the molecular weight of the complex goes up, we get broader and broader line widths, and at some point it becomes unworkable and it destroys the usefulness in the spectra that we've got. Okay, so this is the fundamental limiting feature to biomolecular NMR, which is why we tend to stick with single domains as a preference. So I was struck a few years ago by how elegant Andy Jerome's description of Fourier Transformation NMR was. And so this is lifted straight from Andy Jerome's book. It's a nice illustration if you haven't come across this before. So the idea is if we have a resonator that in this case is represented by a bell, and let's assume the bell doesn't have a whole series of harmonics, it's just a resonator with a single frequency in this analogy. Okay, what we can do is we can ring the bell, we get a sinusoidal oscillating signal that decays away. It has an exponential damping of that as it rings away back to zero. If you run that through the Fourier transform, you extract the frequency component for the resonator. Okay. If you had another bell in the system at the same time, strictly speaking, these lines should be a summation of each other rather than just an overlay. So what signal you would extract at the end would be a summation of those two signals. You run it through the Fourier transformation, and this time it will extract both frequency components of the resonators. And we can add as many resonators to the box as we like. It will eventually get there, a bit slower. And the principle is that if we take a box of resonators and we shake the box up, we record the cacophony that comes out at the end and then pass that through a Fourier transform algorithm, then we will extract the individual frequency components for the original resonators. And that's a wonderful analogy of what's going on in the NMR spectrometer. We use a white light in the radio frequency range to excite all spins simultaneously. We then record the cacophony that comes off of that and pass it through a Fourier transform to get the frequency components in the spectrum. So if you do that to a typical protein, this is a spectrum just looking at a part of the spectrum that is the backbone amide hydrogens for a 10 kilodalton protein. You can see that there is a very limited amount of information we're going to be able to pull out of that because of the amount of spectrum overlap, of peak overlap that we've got in there. So what we can do is go to so-called 2D NMR techniques, and we can spread all of these peaks. This is exactly the same spectrum, but this time what we've done is spread it out into a second dimension. And this second dimension now is the frequency of its directly bonded nitrogen. So we have two axes here. The vertical axis is the frequency of the N15, the nitrogen in the backbone, and the directly bonded hydrogen is shown along the horizontal dimension. This representation of it isn't particularly useful. It's a bit of a cumbersome one to work with. So almost always that is presented as a contour map where we look directly down. Again, this is the same data set, we're just looking at different representations of it. And we're looking directly down on top of it at the individual peaks. And you can see we can spread the peaks out into 2 dimensions very successfully. This correlation, the transfer going from M15 to proton, is something that can be done either through space or through covalent bonds, depending on how you design the experiment. And unfortunately, in an introductory lecture, that's beyond the scope of what we can bring in as to how it works. We just have to accept for the moment, we can generate these 2-dimensional maps in which we're spreading out these peaks so that we can resolve individual sites. Okay, so what I wanted to do then is, as I said at the beginning, we will look at the very first principles of what NMR is about. Where does the signal come from? We've just had a look at, but in what ways does this signal interact with the molecule? And the nicest place to start, I think, is looking at the Hamiltonian operator for the nucleus spin Hamiltonian for the NMR experiment. Now, this isn't put in to put people off with physics. This is put in actually to show how simple the expression is, okay? So this isn't trying to highlight complexity, it's exactly the opposite. So this is describing the energy of the spin system, and it actually breaks down into the summation of 3 terms. There's a shielding part that is determining what we call the chemical shift. There is a J coupling part, and there is a dipolar coupling part. And the NMR spectrum is a summation of the interaction of these 3 components. So what we'll do is look very briefly at the 3 components that we've got. So the first of these is shielding. And the concept is we have this nuclear bar magnet that's sitting in an external magnetic field. So it splits out into 2 energy states. Surrounding that is the circulation of the electrons. And the circulation of those electrons generates a magnetic field that always will oppose the applied magnetic field. So as we have electrons that circulate the nucleus in this way, it will always produce a shielding effect where the nucleus is being shielded from the external magnetic field by the magnetic field of the circulating electrons. Now, anywhere that you get a movement of charged particles, you will have associated with that a magnetic field. And it is this collection of magnetic fields in the molecule that gives NMR spectroscopy its tremendous usefulness. So what we will get for each nucleus in the molecule is you'll get a characteristic frequency in the spectrum. And we'll characterize that by a frequency that's referred to as the chemical shift. And we use chemical shift rather than the absolute frequency, because the chemical shift is the frequency expressed as a ratio of the applied magnetic field. In other words, it is normalized against the applied magnetic field strength. So if we express our frequencies as a chemical shift, every single spectrometer in the world will produce a signal at that same chemical shift value, okay? Because it's more normalized against the strength of the magnetic field that's applied. So what we see is inside any molecule, because there is a complex circulation of electrons that are in there, you'll see an array of internal magnetic fields within the molecule that is dependent upon the structure, that is uniquely characteristic of that structure. And each of the individual nuclei in these positions will sense the local magnetic field at that position, and you can then work back to infer something about the electronic structure within the molecule. Okay. I should say there is a caveat about these fields, the local fields of circulating electrons produce this shielding effect, but we also have a delocalized spread of electrons around pi bonded systems. And these also produce very profound local magnetic fields within the molecule that give the characteristic positions for each of frequencies in the NMR spectrum. If we have none of these magnetic fields in there, then all nuclei of the same isotope would resonate at exactly identical frequency. And the reason that we get a spread of frequencies across there is because we have this local landscape of magnetic fields within the molecule that becomes so useful for telling us what's going on in there. Okay, the second of these effects is so-called J-coupling. This is an effect where what we see is a local field that is produced by the bonding electrons, but it's affected by the coupling of the electrons to the neighboring nuclei. In other words, what we can see is something that is transmitted through electrons that are in covalent bonds. So this effect is always transmitted through covalent bonds. And what it means is that we see a local magnetic field shielding this nucleus that is dependent upon the spin state of the neighboring spin. This can be up to 3 bonds away. As long as there is a covalent bond network between these two nuclei, then it needn't necessarily be a direct 1 bond association. It can be felt up to 3 bonds away. But what you then tend to see is, instead of a single frequency, we actually see the effect of splitting this into 2 different signals that is dependent upon the orientation of the neighboring spin. That is the separation of those 2 signals is related to the geometry and the efficiency of the electron contact in the covalent bonds, and it's always a constant. You can tell something about the geometry in the molecule from that. The third effect is perhaps a little bit more intuitive. It's called dipole-dipole coupling. You have a 2 possible orientations for the neighboring spin, and these will produce 2 different effects, 2 opposing effects on the spin that you're looking at, depending on whether this is aligned with or against the field. Okay, so it's a direct effect, a direct sensing of the spin state of this spin by the by each of the individual nuclei, the neighboring nuclei. So what we actually see is that the separation of these 2 peaks in the multiplet is not just the J coupling, it's actually the J plus dipolar coupling. In solution, for anything any molecule that is tumbling isotropically in solution, what you tend to see is that the dipolar coupling averages away to 0. So the net splitting effect is 0. It has a dependence, an angular dependence on the orientation of this bond with the magnetic field. So if it's turning isotropically in solution with all possible orientations sampled equally, then what you see is the average splitting effect from dipolar coupling averages away to 0. So the separation that you see is actually just J, but strictly speaking, it's J plus the dipolar coupling. Dipolar coupling in structure determination, then, what you see is the dipolar coupling term has an angular dependence to it. It also has a direct distance dependence to it. And for molecules in solution, this averages away to 0. We don't see direct evidence in the spectrum immediately from the presence of dipolar coupling. If we don't exchange the molecule between the different orientations, so in other words, if you got it in the solid state, you will see that we resolve all of the possible orientations for this coupling, and the spectrum broadens out into this blue line. So this red line here is the sort of dispersion that we would normally get for chemical shift range of a proton NMR spectrum. If we have a solid state sample, then the dipolar couplings really are so overwhelming to the spectrum that you lose all fine detail for the resolution of the spectrum. So solid state NMR becomes a problem of trying to remove those dipolar couplings. It's a very different technique to solution state NMR, and it's not one that we do in-house in LMB. we would have to collaborate with people outside, specialists in solid state NMR outside if we were looking at solid state samples. For everything that we do in-house, we stick to solution state. We should say, though, that the dipolar coupling term doesn't disappear. It just averages away. The splitting averages away to 0. But it gives us an effect called the nuclear Overhauser effect, which is a change in energy level populations that we don't need to go into in huge detail. Suffice to say that the strength of the NOE is inversely related to the distance between the two nuclei. So remember, in this case, we've got an isotropically tumbling, rapidly tumbling molecule. So the angular term disappears. So the strength of the NOE largely reflects the internuclear distance, and we can use it as an intermolecular ruler. If we take a sample that has incomplete averaging, particularly if we were to look at the dipolar coupling between a covalently bonded pair. So if this was, for example, an N, a nitrogen atom, and it's directly bonded hydrogen, and we look at the dipolar coupling between that, that would take out the distance term as a variable. And what we would see as the dipolar coupling term would then be an indicator of the bond angle with respect to the diffusion axis of the molecule. So we have two really useful properties for structure determination that come directly from dipolar coupling. One is the nuclear Overhauser effect, and the other is this residual dipolar coupling. Nuclear Overhauser effect, then, it comes really from the 1980s. It was spearheaded from the 1980s by Kurt Wütrich's lab in Zurich. And let's get rid of that. And the structure, the solution structure of a molecule is determined from measuring specific HH distances in a molecule. So if you look at all of the hydrogens that are present in a protein molecule, we will obtain the frequency assignments for each of these hydrogen spins individually, and then we can look at the strength of the NOEs between them to determine what the distance are. Typically, we would obtain something like 25 restraints per amino acid residue, and that gives you a good handle on a high resolution solution structure. So this is an example of a two-dimensional NOESY spectrum, NOE spectrum, if you like. The idea here then is that we've got two frequency axes. A peak here, for example, is saying that we've got a spin that is at that frequency, is very close in space to a spin that's at that frequency. And because we know the assignments of each of these, we know which two nuclei that's referring to. From the strength of that, we can correlate the strength to the internuclear distance in between them. And as you can see, there are thousands of NOEs that we can measure within a molecule. It's an extremely complicated process to analyze, and it takes a long, laborious amount of time to do it for proteins of any size above about 15 kilodaltons or so. It becomes quite a slow process. This is why it would be preferable speed-wise, if you can, to crystallize the protein and get the structure that way. But if you need it, we can also do it. And just in this last year, we've done a couple of formal high-resolution solution structures around for groups in LMB for proteins that won't crystallize. The quality of this spectrum degrades as we go up in molecular weight, and realistically, the molecular weight limit for a high-resolution NMR structure is around about the 40 kilodalton marks. Higher molecular weight systems have been looked at, but that would certainly be non-routine. So realistically, we're looking at a ballpark figure of about 40 kilodaltons. Residual dipolar coupling, we're saying just a little bit about that. So what we record is the splitting. So this is, for example, a proton signal that is split into two by coupling to its directly bonded N15. We record that for the isotropically tumbling molecule. If we then put the same protein into a solution that is collimated with respect to the magnetic field, we can place a small degree of alignment so that the molecule doesn't average fully over all possible orientations. If we're careful about the way in which you put the alignment in, you can get a situation with about 1 part in 10 to the 3 excess population of the major diffusion axis aligned with the B0 magnetic field. That recovers us a few hertz of dipolar coupling, referred to as a residual dipolar coupling, that then increases the separation of these two spins. So the dipolar coupling value is the difference between this splitting and the J coupling that we see for the isotropic solution. That might look quite underwhelming as a first look, but if we look at a simple example of that for a 3-helix bundle, you can see that the NH bond in each of the helices is roughly, approximately coaxial with the helix axis. Okay, so we would expect that each of the helices shows the same orientation for each of its NHs. And you can see in this plot of the RDC value that we get as a function of residue number, that we get a good correlation in the orientation of helices within these 3 different planes. So it gives us an ability to align these helices with respect to the diffusion axis of the molecule. We can do that in a slightly more advanced way by recording other dipolar coupling values as well. So we can do one for the H alpha and C alpha coupling, etc. So we can get this H alpha C alpha bond orientation, the NH bond orientation, the C alpha C beta bond orientation, etc. You can put those together to get a unique orientation for each peptide plane in the molecule. This is extremely useful for looking at the orientation of intrinsically disordered proteins and in IDP segments within larger protein assemblies. This is P53 full length. It's about 355 amino acids as a full length protein. It has 4 different subunits here. We have modeled on 20 different chains for each of the 4 N-terminal segments that have residual ID, have intrinsically disordered structure. And it shows the degree of disorder in there. But when we run the RDCs on this, we can see that the experimental data in blue only fits the simulated data when the simulation includes a single turn of alpha helix at one of the recognition sites, the known binding recognition sites, and it also contains about 22% polyproline helix in the rigidified C-terminal end of this extension. So it gives us a good handle into how the molecule is behaving in a complex situation. The last thing I will say, quite briefly, is the process of spin labels, the use of spin labels in NMR. So if we put a paramagnetic radical into the protein at some point, then it has a catastrophic effect on the relaxation of spins within about 35 angstroms of it. Certainly, perhaps we should say catastrophic up to about 20 angstroms, and beyond that, it can be detected up to about 35. And what happens, it just causes everything to relax extremely quickly. We can use this to measure distances in proteins by introducing a synthetic label. Tempomylamide attaches in a facile coupling reaction to cysteine thiols. We can then reduce this with ascorbic acid to a diamagnetic hydroxylamine, which then we can use the spectrum of that as a control. So you compare the magnitude of the spectrum and the relaxation rates that you get before and after the addition of ascorbate, and this has extremely useful consequences for being able to characterize bits of distances within protein complexes. So it's, I should say in there, it can be used for detection both transiently and stably populated states, also looking at intra or intermolecular proximity. So once we've got all of these restraints that we put together from the NMR, we put it into molecular mechanics, standard molecular mechanics force fields. So everything is treated, all the bond lengths and bond angles, whatever, are treated in the same manner as a Hooke's law spring. So we simultaneously optimize all of the coordinates within there in the molecular mechanics package, and into that we will add experimental restraints from the NMR data. So the experimental restraints, just to reiterate, is nuclei over Hauser enhancement, residual dipolar coupling for the bond orientations. We can use chemical shifts to give us some idea of bond dihedral angles. Spin coupling constants gives us ideas of geometry. We can put in hydrogen bonds that we can measure from hydrogen exchange protection factors, individual per residue protection factors that we can measure, and this paramagnetic spin labels if we need to get in there. Add all of those together with the molecular mechanics force field, and you get a high resolution structure that is completely consistent with all of the experimental restraints. The overall process, it's worth saying something about here, is that we take a molecular coordinate, geometric coordinate, and we minimize the energy of that system, right, by simulating that we start off at high energy in the system, putting high amounts of thermal energy, allow everything to shake about and allow exploration of different conformational space. As we withdraw the energy from the system, ideally what's going to happen is it's going to sit eventually in the lowest energy state. In practice, we are simultaneously optimizing thousands of different molecular coordinates, conformational coordinates. So it's inevitable that we can get trapped in local energy minimum rather than the global state. So the way in which we can be sure that we've got to the global minimum overall is by starting the calculation from 100 different starting points, taking the energy out, and then seeing that they all converge to the same point. This is why when you look at a protein data bank and you download a crystal structure, you see a photograph of a crystal, a single state. If you download NMR structures, what you see is an ensemble of overlaid structures, and you're looking at the convergence that you've got from the different annealing states. So typically when you download NMR structures, they're not a single structure that you download. You will get an ensemble average. Okay. We won't say anything much about this, just that the spectrum, as we might expect, a proton spectrum of a protein is largely breaking down by the primary structure, the electronic structure around each group. So each of the HNs, for example, is resonating in a narrow region, and similarly methyl groups in the side chain will all be in a fairly narrow region. So the spectrum breaks down according to the composition of the molecule. I should also say that we need to take steps to suppress the water signal, because water's there at 55 molar, so 110 molar water protons, and we have proteins that are generally there at something like 500 micromolar as a maximum. So we need to take efforts to suppress the water signal. The standard spectrum that we look at is the so-called HSQC. It stands for heteronuclear single quantum coherence. Right? And in order to get one of these spectra, we must specifically introduce N15 as a stable isotope label by growing the protein in a medium that contains N15 ammonium chloride as the only nitrogen source. Okay. Without it, then we don't see anything in here. So we can put in binding partners, for example, and you don't see any evidence of the binding partner. You don't see interfering signals that come in from the binding partner. The only thing that you see is the signals that come from your labeled protein. And what we have is an individual spot in there for each of the NHs in the molecule. So effectively, we have a probe, an individual probe for the structural environment at 200 different sites in this spectrum. And we can use that to tell in a very detailed structural characterization where there are changes, structural perturbations occurring in the molecule. So for example, we can titrate in a binding partner. You look at what happens to the chemical shift changes. Anything that's outside of a binding site, it doesn't change. But for the resonances, the spins that are in the binding site, we see a migration as we go to increasing levels of the binding partner. We can then map those chemical shift changes onto the surface to get a heat map of where the binding interactions are, binding interfaces are. This is quantitative, and if you do a whole series of titrations and plot the chemical shift change as a function of ligand concentration, you get a definitive value for the dissociation constants and equilibrium constants for that. It doesn't have to be, that was a fairly straightforward, simple example, but we can also look at much more complicated examples. I'm not going to dwell on this too much. It's not particularly easy to see, but we've color-coded, so this is from Stefan Freund from a few years ago. He has color-coded the structural changes that are occurring, the chemical shift perturbations that occur in a multi-liganding event for looking at blood clotting proteins, the activation of blood clotting enzymes. And this is 3 different ligands to the same protein, and you can look at the structural consequences of debinding of each of the individual sites. So you can dissect with real clarity some complex binding equilibria. We can then not only tell where a binding interface is, we can use modeling algorithms such as Haddock, developed in Utrecht University, for getting a high resolution complex. In this particular case, what we've done is we've used what's called a half filtered NOESY spectrum. So we only allow the signal from protons that are coupled to a carbon-12. So in other words, they're not coupled to a C13 in this dimension. And we can apply a second filter to only detect protons that are coupled to a C13 that's introduced into the molecule. So if we take a C13 labeled protein and an unlabeled peptide, essentially what this protein is, this spectrum is showing you is the NOEs that are filtered out solely as coming from the peptide onto the protein. So it's across the interface as the NOEs that we get in here. And we can assign each of those. In this particular case, we look at the shift perturbation for this coupling complex, which is colored in orange. We then add into that 35 intermolecular NOEs that are going across the interface, and we apply this Haddock flexible body docking to get a high resolution model of the complex at the end. We're asked, what is the molecular weight limit for NMR? The answer is there isn't really a hard and fast molecular weight limit. If you want a high resolution structure, as we've said, an NOE-based structure, around about 40 kilodaltons, above that is going to get exceedingly challenging. In practice, though, even for something that's a quite large complex, like 255 kilodalton complex, we can still see if there is perturbation in the spectrum as you add a binding partner to it. So you can still get some useful information. We're not going to get full assignments for that and work out where the interfaces are, but definitely we can report back that we get intermolecular associations, and we could measure binding constants and affinity constants for that on a large complex. So it's really not straightforward to answer the question, what's the limit? You can go and get information from very large complexes. And here is another example of looking at large complexes. We don't have to be fixated with looking at the signal of the macromolecule itself. You can look at the substrate. So in this case, it's a recent one from Jan Lerver's group. Nicolas Jean came across, and we looked at ATP hydrolysis, the rate of ATP hydrolysis. You can pull out kinetics information, and in this case, partly to look at the structural integrity of the protein. Under what conditions was it still turning over substrate? And that can be done by direct observation of the substrates or metabolites that are in solution. So it's worth bearing those in mind. The full molecular weight of this is over 200 kilodaltons. We can also look at complex mixtures Even from the 1970s, there were attempts to look at whole cell NMR, and you can look at cytoplasmic contents. So we said this earlier on, that if we were to take a proton spectrum of an intact erythrocyte, human red blood cell, then we will get a spectrum that is showing signals of the macromolecules and signals of the small molecules. And surely it couldn't be simple enough, could it, that if we were to delay the start of acquisition for a short period of time, so we don't acquire signal during this point, then this signal will decay away. This is a signal from a macromolecule will decay away, and we'll lose almost nothing from the intensity of the small molecule. Well, it is almost that simple. And by applying a so-called relaxation filtered experiment, we can get high resolution spectra of the internal contents of the cytoplasm. And we can follow individual biomarkers. There are groups looking at several hundred individual biomarkers at the same time, simultaneously, as part of metabolomics studies. We can tell the molecular weight, the effective molecular weight of a protein. This is not a mass spectrometer, but it certainly gives you enough resolution to work out if you're looking at monomer, dimer, trimer. And that can be done very easily by the ratio of relaxation rates that you see. I won't go into too much detail in there, but we can get certainly resolution as to the polydispersity of protein samples. One of the things I would put across to people is the idea that protein NMR, by observing the protein itself, has a built-in advantage that you can always see if the protein's gone off. So you have this built-in quality control check. Is your protein folded or unfolded? Well, you'd see it instantly, okay? And if during the experiment, the protein goes off and you're no longer working with viable protein, you'd see it. Basically, in this case, all of the signals from an unfolded protein will be exposed in the same solvated environment in solution. They'll have very uniform environment. If the protein is folded up, then all of the NH is spread out into a whole array of different environments that give them this extra signal frequency dispersion. Okay. So we can see that very well for the NH is also up in the methyl region. This is all of the methyl groups. The two proteins here are of the same molecular weight. One is unfolded, one's folded. And these methyl signals in a folded protein get dispersed out in their frequency, and it's very easy to see as a protein quality control check. In two-dimensional spectra, it looks something like this. A typical folded protein looks like that. Unfolded, you get very narrow dispersion of the amide protons, and an aggregated protein would obviously show up as being quite an ugly looking spectrum, but exceedingly useful as a quality control check. Per residue dynamics, we can encode into the HSQC. So we use a modified HSQC experiment in which we can encode the dynamic behavior, basically the mobility rate, into the intensity of peaks in here. These blue peaks here are negative, and if we plot out the intensity of these peaks, we can see that there is a correlation between chain rigidity and the magnitude of this NOE between the proton and the M15. But crucially, we're getting this information on a per residue basis across the whole chain. That might look really trivial and unexciting, but this is a whole field of study in which you can look at multi-domain assemblies. This is a 3-domain protein, a clinically important sample from Alan Warren's work that Stefan Freund was doing a few years ago. What we can see from the relaxation rates that we've obtained from these HSQC spectra, you see that one of the domains is rotating at a very different rate to the other two domains. So these two domains are rotating together as a single rigid body, and the third domain is different from that. Some of the clinically important mutations are linked to mutations in the linker region that affect mobility. We can also illustrate in here, we've got the comparison of human and yeast homologs in here, and you can see that there is one loop region, specifically in the yeast, that has a very much higher mobility in the yeast homologue than it does in the human. So this is the sort of per residue level of resolution of what's going on in the dynamics of a backbone is exceedingly useful information. We can look at individual chemical exchange processes. Chemical exchange, in this case, it could be anything. This particular example is looking at the pKa individually for different side chains. So we got 6 different acidic side chains that we can titrate as a function of pH, look at the change in chemical shift, plot that out, and you get an accurate pKa value in there. It could be also kinetic resolution of phosphorylation sites, for example, looking at where different kinases are phosphorylating. We could be looking at site-specific changes and perturbation, protein folding and cofactor turnover, whatever. But the important bit is it's quantitative and it is site-specific. I'm not going to go into this a bit over time, and we can say a little. Well, we could say a lot about NMR screening applications for the pharmaceutical industry and looking at small molecule ligands that are binding to protein surfaces and looking at libraries of small molecule compounds and pulling out those that bind to the protein surface. It breaks into 2 different categories, which are the ligand observed or the protein observed analyses. The advantage of protein observed is you get that protein quality control check. If the protein just crashes out of solution, you would see it. Those are different approaches for ligand observed methods that all give complementary information as to whether the molecule is binding or not binding. So in this case, we've got a mixture of 2 compounds, one that binds, one doesn't, and we can distinguish those easily by complementary NMR techniques. So just to summarize then, the advantages of what we can do, the scope of biomolecular NMR is looking at the protein quality control, so we can do verification of structural condition. We can look at 3-dimensional structural characterization and looking at perturbation in the regions of structural characterization, including the formation of complexes. Dynamics and looking at the internal mobility from relaxation rates, we can look at diffusion and conformational exchange processes. Biomolecular interactions is a key theme of what biomolecular NMR is about. And in all sorts of classes, and we can produce quantitative data for biophysical constants at the end. So that is the last slide, which is just recommended. I think this goes online on the LMB website. So if you want to look at further reading, then there's some suggestions of where you can go. If you want to know more about the physics, the spin physics, please do come and talk to us. There are people in the NMR facility who are there to collaborate on any of the projects throughout the divisions of LMB, and you're encouraged to come and see us to talk about it, even if you're not sure whether this will apply to you or not. We can certainly advise you on anything that would need doing. If you want to know any more details, either come and ask us, or you can start on one of these texts in the first place. So thank you for listening. And Jane will be giving her talk on advanced applications of NMR on Thursday morning.
So if we have any questions for Trevor, could you type them in the Q&A, please? And okay, we've got one. I'll let Trevor read them out and address the question.
What strategies do you use to study concentration-dependent oligomerization? Yeah, so the 2 that we would go for generally are quite quick experiments. So you can either look at the rotational diffusion of the molecule, which is the example that I showed in there, or you can also look at translational diffusion, which is so-called DOSY experiments, if you may have come across those, where you can look at literally the translational diffusion rate, which correlates with molecular weight for folded proteins. Either of them give usually similar answers. The so-called spin echo that is looking at rotational diffusion, that takes usually about 10 minutes for us to get. So it's a really quick assay on an NMR timescale. A lot of experiments are running for hours or even days, but in 10 minutes you can usually tell within about 2 or 3 kilodaltons resolution what the average molecular weight of your complex is. The diffusion measurements take slightly longer, maybe half an hour or so, maybe an hour. But those are the two procedures that we would go to as a first point of call. If there's no other questions, we'll wrap it up there then.